Hello, everyone, and welcome to today's webinar, Single-Cell Antibody Discovery Using Islands of Automation. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, please visit beckman.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I'd like to now present today's speakers, Paul Anderson, Senior Research Scientist at the Lilly Biotechnology Center in San Diego, California. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Paul, you may now begin your presentation. Welcome.
Okay, well, thank you for the introduction. My name is Paul Anderson, and I'm proud to present the work that we've been doing at Lilly for the last several years in automating a single-cell process used for biologics discovery, specifically antibody discovery. I joined Lilly 2 years ago, so a lot of the work I'm sharing today is the work of a fairly large group of people, and some of the people that helped make this presentation and then had a major role in developing these systems are my co-authors, Mike Cohen, Melissa Crisp, and Tom Kujek. So I just want to thank them for the opportunity to share their work and the group's work today with everybody. So I'm going to dive into talk about the process that we're automating here. We have several antibody discovery platforms at Lilly, and the one specifically I'm going to touch on today is immunization-based platform using single B cells and going through a series of automated platforms to generate lead molecules. We have several other platforms, as I mentioned, that are orthogonal to this approach that we use for antibody discovery as well. So this is the main process I'm going to be talking about is this single B cell application. But keep in mind, a lot of these systems are used in different aspects of other biologics discovery efforts. And so this presentation is really just honing in on this application here. So we have several platforms I'm going to discuss throughout this process. And these are really all tied together with a LIMS infrastructure. So we are working on collecting information from each of these platforms in the form of data, as well as sample tracking. And so really one of the overarching themes here is the software side of the process, where we're capturing a significant amount of data, whether it be purification amounts from DNA or purified material amounts from expressed antibodies, data and associated with titers, and a significant amount of screening results toward the end of the process. And what we're trying to hand over is really a data package so our project teams can make informed decisions and triage down to lead molecules as fast as possible. So our process, this particular process starts with the immunization-based strategy, as I mentioned. So that could start with mice or a variety of different host organisms or host species. In this example, we're using a mouse model, and so we would immunize that mouse with the antigen of interest. And then once we have sufficient serum titer, we would isolate the B cells and use a back sorter to dispense antigen binding B cells into 96 well plates. And so we typically start with roughly 20 96 well plates. So the process I'm going to talk about here, usually it's about 20 96 well plates, so about 2,000 single B cells will go into this process as this first pass. several projects where we go through multiple passes, several immunization strategies, and so they grow significantly larger in size. So first system I'll talk about is what we call the fill and barcode system we developed with Beckman. And this was essentially a automated liquid handler with a series of other third-party devices on there, such as a Cytomat, a labeler, a sealer. We added a balance recently. And really this system is designed to add reagents to plates, barcode or label those plates, seal them up to feed several platforms throughout our process. With the case of where we're sorting cells into plates, we typically are using, those plates are created on here with cell sort buffer and sealed up and stored in a chilled manner until we're ready to sort cells into them. Then once we sort cells into these 96 well plates, they come back onto this platform when our scientists are ready to process them, and then they are consolidated into 3D 4-well PCR plates. And so the next system I'm going to talk about, where a lot of our molecular biology takes place, this system feeds that as well, adding different PCR master mixes to a variety of 3D 4 PCR plates. And so the plates come off of here. They've been consolidated into 3D 4-well plates, and they would come to what we call a molecular cloning system, also developed with Beckman. And so on this system, I'm going to touch on some of the usual suspects of instruments that you'll see on several of the systems coming up. And so that would be a plate lock and X-peel, and those are for sealing and peeling of plates, as well as a V-spin, a centrifuge. And so that is typically to eliminate bubbles and to pull samples to the bottom of a well. And there's several Cytomats on our system. So we have an ambient Cytomat on this system, as well as a chilled Cytomat. So as we go through the presentation, I'm really going to try to touch on what makes each of these systems unique in their own right. So the molecular biology, or the molecular cloning system, the kind of special sauce on this guy is the bank of automated thermocyclers that you can see in the top. So there's a stack, 3 stacks of 4 thermocyclers, so a bank of 12 automated thermocyclers on the system. And this is what we use to carry out a series of PCR reactions on here, where we start with the single B cell sorted into each plate. We split it to heavy light chain and assemble it into our vector of interest. So the output of this system is going to be heavy and light, a set of heavy chain plates and a set of light chain plates assembled with the antibody gene of interest. The first QC step in our process incorporates, on the top left here, you see an ATI fragment analyzer. So we cherry pick samples after the second round of PCR, and this really, we run these on the fragment analyzer, and this lets us get some insight as to, are we chopping out the fragment of interest and isn't it the right size? And so this is, usually we have a fairly high success rate of this, but if there's anything going wrong with our master mixes or anything went south in the sorting side of things, this is where we would typically catch problems. And so as you come off of this system, we have our heavy and light chain DNA. We go into a semi-automated E. coli transformation step. And so we, in a 3D 4-well manner, do a transformation into competent cells, let that sit for a bit, and then transfer those into 96-foot deep well plates with LB broth. And those will sit in shaking incubators overnight. And after that incubation overnight, we pellet the cells, we centrifuge and pellet the cells and decant them and seal them and store them at -20 degrees until we're ready to do our plasma purification. And so the next part is we built a recent system to do our plasma-based purification. And we are using the 96 turbo kit from QIAGEN. And so what you see here on the top left is the E. coli pellet block. We have to dispense a few different reagents to resuspend, lyse, and neutralize the E. coli. And then we transfer that material into a lysate clearance filter plate, and samples go through that to clean up the garbage that's in those samples, it's kind of snotty, viscous material. And it gets eluted into a DNA binding filter plate. You pull samples through that filter plate, DNA binds to the membrane, do a couple rounds of washing, and then elution to purify to do your plasma purification. And so as we approach automating this, our manual workflow involves vacuum manifolds, and after this last wash step, we would do a centrifugation step to really give a hard spin to these filter plates to get rid of any residual wash buffer before we do our elution. Automated vacuum manifolds are quite common, but there are significant drawbacks when you have samples that could be, if you have certain wells that don't have samples, the vacuum is going to pull through the path of least resistance. If you have other wells where that are slow or clogged a bit, then those tend to not get pulled through. Once you have wells around them that get pulled through the vacuum, the ones that are clogged or running slower tend to be more and more difficult to get to be able to pull through the filter plates. And so we, in talking with Beckman, we had explored some other options with how to proceed with handling filter plate-based purifications. And so we had the idea to explore positive pressure devices. So we had this discussion with Beckman. They talked about some positive pressure devices, and then we started looking into it more and found several options on the market. And so we brought a few different devices in that I'll talk about on the next slide. But to just give you a little bit of a background on what these devices are and how they differentiate themselves from vacuum and centrifugation. The way they work is pulling in house air. It could be a gas, nitrogen, or different type of gas into a pressure reservoir. So you have a high pressure side on the reservoir, and it uses a flow restrictor, and this is essentially software controlled to open up small holes and let some of that gas or air through. And so the nice thing about this, you're essentially pushing samples through a filter plate. And whether you have an empty well or a well that's completely clogged, since you have a large pressure differential across the restrictor, you have the same amount of pressure being applied to every well. So whether it's empty sample or a clogged sample, you don't have those wells impacting the nearby wells or adjacent wells. And so it's a very nice, consistent flow rate across the plate, which is highly desirable. And the other thing is, essentially, to get into these positive pressure devices, you're stacking and unstacking labware. And so that's a big benefit as well, because that's a fairly simple mechanism to automate. So while vacuum is very inexpensive and easy to automate, it has some quality and reliability issues that we would be concerned about. And so the centrifugation method, you do get even flow across all samples, but is a bit more challenging to automate stacking and unstacking and the waste associated with plates going in and out of the centrifuge. And so that was a bit more of a red flag for the centrifuge-based method of handling filter plates. Whereas positive pressure, it's a bit more expensive, significantly more expensive than vacuum, kind of on par with automated centrifuges. But it's very fast, easily automated, and the flow control is very good. And so we opted to explore this route a bit further. What we did find in our due diligence is that this is essentially this is very new for most vendors. And so the ones that we reached out to, they were just starting to automate these types of devices. And so we brought in 3 devices from 3 different vendors. And what I'm showing you on the top right here is in blue our heavy chain DNA and in red our light chain DNA. And so we have control samples which would be done manually using our vacuum and centrifugation workflow. And then all 3 devices performed well in our hands. And so you can see there's a bit of a bump in DNA concentration with the heavy chain and a significant increase in DNA concentration with the light chain plates. This was I would throw out the disclaimer that this was a fairly quick and dirty experiment using these 3 different devices. Not a lot of optimization done to really dial in flow rates and dial in other conditions related to these devices. So as a first pass in some early data, we were very pleased and encouraged to explore this technology further. What we did as well, I mentioned in the first system, we have automated balance on there, and you'll see that throughout the systems I'm going to talk about. We weighed the filter plates before and after each of the liquid handling steps. And so to explain the higher concentration on here, what we think is happening is after you do your last wash step, when you are pulling that through a vacuum and then going to a centrifuge to spin out as much residual wash buffer as possible, there was, based on gravimetric results, more wash buffer left on the filter plate than when we were using the positive pressure devices. So we believe that we were effectively eliminating, pushing through more wash buffer, more residual wash buffer on the positive pressure devices. And then when you go to your elution, you've reduced that residual wash buffer more effectively on the positive pressure devices. And so our elution had a higher concentration of DNA. So that's really what we believe to be the main cause of the increase in concentration. And so we did look at the quality of DNA. We ran it on gels, and I'm not showing that data, but it was right on par with the positive pressure device and our normal workflow. And so we believe the quality of the DNA was good. And in the bottom right figure, I'm showing expression titers. And so what really matters at the end of the day for us is how the yields look. When we take the antibodies, the DNA that's purified on these, and we go into transfection and look at what the antibody yields are. And so black bars are a couple replicate plates, and then red, purple, and blue are the 3 different vendors, positive pressure devices. And there is some variability in here, but by and large, we have run-to-run and plate-to-plate variability that's within this degree on our transfection system. And so we believe this, the DNA produced from these positive pressure devices was right in line with what we were looking for. And so it was nice to see those results. And so we opted to go ahead and work with some companies and ended up working with Beckman to design a DNA purification system. And so the first system that was proposed was a Biomech i7 with 2 positive pressure devices, and they partner with Amplius for these positive pressure devices mounted off the side of a Biomech i7. And then we have, again, plate lock, XPO, multi-flow for the reagent dispenses, and some bioshakes on the system as well. And we have a reader on here. So as we purify DNA, we can go take a aliquot and measure it and determine what the purified yields are. And so what became apparent as we first designed this system and looked at what it would take to run 40 96-well mini preps on here. What I mentioned earlier was we started this process with 20 96-well plates of sorted cells, and those get split into heavy and light chain plates. And so that's where the 40 96-well plates are coming onto the system. So when you look at the plates that are coming in, the tip boxes required to transfer samples from the E. coli pellet deep well plate onto the filter plates themselves, elution plates, and tip boxes to transfer to make that aliquot of eluted material. There's a lot of labware you can see associated with this process, as well as looking at the time in the models with Beckman was 11.5 or so hours just for the DNA purification side of things. And so this led us to start exploring another bottleneck on here, which is all the labware that we have to have on this system. And so what we took a look at was the Thermo Skyline that had come out right around the time, or just before the time we were starting to design this system. And so this can be thought of like 14 PEZ dispensers. And so without having shelves and you don't have a random access like the standard Cytomats, you essentially can stack up filter plates, stack up your tip boxes, and really have a high density storage device on here. The other thing I mentioned is we have a reader on here, and we are able to measure how much DNA we've made on here. Well, the next step is to normalize and mix your heavy and light chain DNA, and I'll talk about that in a little later in the talk. But since we have the information on here and we have a liquid handler on here, we have all of the tools in place to be able to do that mixing. And so we modeled that up as well. And so that's where you see additional labware on the right. So we have another set of tip boxes. We have the mixed DNA plates, which should be 96 deep well plates. And then we make an archive copy, which would be the 384 well copy with a small amount of DNA, and that's kind of like our insurance policy if anything goes wrong with the original plates that we've made. And so when we model the DNA purification with the mixing, we're looking at roughly a 20-hour workday or 20-hour automated process to go from E. coli pellets all the way to DNA that's mixed and ready to be transfected. And so that was it doesn't leave a lot of time if you're going to do this in subsequent days, and a lot of this is going to be running at night. And so what we wanted to look at, we're also exploring a couple new devices on here, the positive pressure devices as well as the thermocytomat Skyline. And so to de-risk ourselves a little bit, we worked with Beckman, and they had a nice proposal on upsizing this system a little bit. So what we have here is what the final system layout, and it's another i7 with 2 more positive pressure devices. And what you see are the Skylines are mounted directly underneath the Biomech i7s. And with all the labware that we're moving around, the robot movements start to become one of the bottlenecks on here. So by mounting these Skylines directly underneath and having the Biomech pull tip boxes and filter plates directly out of the Skyline and move on to the deck, we're minimizing transport times and speeding up the system significantly. And then having the redundancy of essentially a mirrored liquid handlers, mirrored positive pressure device and purification systems on here gives us a bit of an insurance policy as we, anytime you're automating new types of instruments, whether it be the thermocyclers or the fragment analyzer, there's always unforeseen things after you go through a significant number of samples over a couple years. You start to find what things work and what things are a little bit not as reliable or robust. So by having some redundancy on here, we really are happy that we have some insurance policy to keep the system running at all time. And so we've put a balance on here as well. The balances are throughout the systems you'll see are really to do some automated verification. So as we tell our liquid handlers to dispense a certain volume or to transfer a certain volume, we were weighing most of the plates through all our systems before and after each of these steps. And this is our QC step to make sure our liquid handlers are holding up as expected. And so we have the positive pressures in place. We have high density storage using the new Skylines. And so with the new model for this system, with some parallel processing between the two wings of the system, we can get the purification down to a little over 7 hours and then mixing in about 4 hours or so. So this is pretty reasonable for the number of plates we're putting through the system, a little over 11 hours for a full purification and mixture. And so from there, we go to the protein expression system that was developed a few years back with Fujifilm, or Waco at the time, and now Fujifilm. And so we take those 96-well plates with normalized and mixed DNA, we put them onto this platform. We add our transfection reagents and cells, and then go into these Cytomat 2C tower shakers. And so these are shaking incubators. And some of the nice features of this system are we have 4 Cytomats that each have a 14-plate capacity, so 56 total plates can run on at a time. Again, when we developed this system, these tower shakers were early days, and so by having a bank of 4 of them, that's been a nice insurance policy where we've had issues that pop up from time to time. We're able to continue running. Typical runs are in the 20 to 40 plate range, so we're not using all 4 at a time for most runs, and so we're not using all 56 positions or all 4 Cytomats at a time. And so we've been able to get by and keep our process moving forward by changing the configuration of these on the fly and using as many Cytomats as needed for a given day. When we go through our process, right now I'm talking about a 96-well process, and we're doing close to a little under 1 mil expressions on this system. We eventually get to a point where we triage these samples down and people need more material, and we'll purify samples. And so what runs on here is not just 96-well plates, but we also do 2 and 4 mil transfections and 24-well plates. And those are all run in one run on here, and based on the barcodes of the plates, the system will recognize whether they're 24-well samples, 24-well plates with 2 mils or 4 mil samples, or 96-well We call them 1 mil, but they're a bit under 1 mil in total volume. And based on the barcodes, the system puts the plates on the different liquid handlers to add transfection reagents and cells, and it uses the barcode, a suffix on the barcode, to determine what method to run for that plate. So we treat all the plates as the same and set the system up in an identical manner, and then the system applies different methods based on the actual plate type it's running. So that's a nice feature of the system. And then we recently added a balance to this system, again, for automated liquid handling verification. And then there's a dispenser on here for some reagents that has a nice manifold system. So we have a very viscous solution on this as part of this workflow. And so it does an automated cleanup and washing of that dispenser after hours. And so that's a nice feature of the system as well. So our DNA gets put on here. We add our transfection reagents and cells. And typically the next day they're getting pulled off of the system and put into Kuhner shakers for roughly a little under a week expression. And so as they come out of that week expression, then we put those plates with cells that have been, CHO cells typically expressing the material over that 6 to 7 day timeline. And then we'll go on to what we call the Beckman quant and cherry pick system. We typically refer to this as our IgG system. And so the workhorse instruments on here are the Bionics High-G centrifuges. So these are really nice centrifuges. So if we have different volume ranges, and the rotor and the balance tolerance for these centrifuges are very excellent, and we can spin them at very high speeds reliably. And so we decided to upsize the centrifuge on this particular system for that reason. So we have the Bionics High-G centrifuges. We have a fleet of NXs on here. So we pellet the cells in the centrifuge, and then we use the Biomech NXs to transfer supernatant into a fresh plate and take a little aliquot of that into a 384 tilt welt plate compatible with the octet. So we were one of the early folks, obviously with Beckman and in collaboration with Forte Bio, to integrate the octet HTX. And so we take the aliquot of the supernatant, put it on the octet HTX, and use the octet to measure our titers from the crude supernatants. And then that information, using some scripting that was developed with the Beckman folks, we are typically setting a cutoff for what we call an express sample in around 5 micrograms per mil. And then we'll go to one of the other NXs and then cherry pick samples that are above that cutoff, and then leave certain wells or column empty for controls for downstream assays. And so we're typically measuring titer on the octet and then cherry picking and rearraying the samples on another NX on the system. And again, we have a recent balance on here. We have a barcode labeler, so as we make new plates, we're pinging our LIMS system and then coming up with new barcodes and telling LIMS system at the end of the process what these new plates are, where the samples are, and what concentration material they have in them. And so it's a really nice platform that's been really one of the workhorse systems for this and several processes in our discovery efforts. And then I'll talk a little bit later. We recently put some bioshakes on here and where that comes into play. So from this system, we essentially have supernatants of a good amount of expressed material that we're ready to start testing. And so what we have in place, and what we had in place, was these plates that come off the system, and they're variable concentration supernatants, and these values were determined on the octet. And then we would determine what our screening plate setup would be, and we would typically need to normalize these variable concentration samples using a Lynx, which is a 96 variable volume pipetting device, to a flat concentration, say 25 micrograms per mil. And then from there, we would go into Tempo software that drives our Echo Access, our lab site access system, and make dose response plates that would be used for screening. And so really the only reason that we're doing this normalization step is because the software that drove this system, it was largely built for the small molecule world and assumes that the plates coming in are all of a uniform concentration. And so we wanted to explore options to go from a variable concentration starting plate to get to the same dose response plate with a software-based solution. And so we work with the folks at Biocero and incorporated their transfer map work list generating software and the scheduler Green Button Go to drive this system to do exactly this. So we can now take the variable concentration supernatant material, take the same type of dose response plates, and then generate work lists and feed them to the echo to accomplish this task and eliminate that normalization step. And so this is something that's been in place for a good chunk of this year now, and it's been working nicely. So it's a nice example of a software solution to really streamline our workflow a bit. So once we have our dose response plates ready, then one of our recent systems late last year we affectionately call the Beast, and that's a bit of a play on Beckman assay system. And so it's the drawing doesn't quite do it justice, but the scale is far larger than all of our other systems. To give you a bit of a flavor of the scale, this is a 1.5 meter precise robotic rail on the right, and on the left it's a 2 meter rail. And it's a fully enclosed system with 25 HEPA filters on top, capable of running BSL2 screens. So I'll start kind of in the top left corner and go around the system a bit. So we have two GNF WD2s. They're mirror images of one another, flanked by a stack of Mettler balances, again for liquid handling verification. I'll talk a little bit about that in a bit as well. We have a suite of liquid handlers, including the WD2s, and we have a Certus over a Combi, as well as two Blue Cat blue washers. And then we have, again, the usual suspects, plate locks, A4S sealers, XPLs on here. We have a couple biotech spectrophotometer readers, a multi-mode reader from biotech as well, the Synergy Neo 2, as well as a high throughput flow cytometer, the ZE5 from Biorad. And then we go around to a Biomedical i7, and you'll see a couple of positive pressure devices off the side of that, and I'll talk about a little bit later on. And the i7 is outfitted with the 96 multi-channel head and a span 8. And then we have the i5 that has a 3D4 head on there. And then a couple cytomats, typically one's always at 4 degrees and one's at 37 degrees. And so it's really, you can see most of the systems I've discussed thus far are built around doing a fairly fixed workflow. This is the kind of jack of all trades where we're going to run everything from ELISAs to cell-based assays to high throughput flow assays. And it's large and it has a lot of equipment, but no screen incorporates all the equipment. You basically are looking at what you want to accomplish with a specific screen and applying the best tools for the job. And we have a lot of tools on this system to be able to accommodate any type of assays our scientists would like to run. And the one thing I want to talk a little bit about is how the balances are incorporated onto this system in particular. And so we have balances that work with our WD2s to have a feedback loop to control dispensing. So when we got this system early on, I put a 2-liter bottle of PBS onto several of the different types of dispensers on the system and just dispensed 20 microliters per well into 3D4 well plates, and repeated that for hundreds of cycles. And what you see here, this is normalized to 20 microliters. And the theme of this is basically all the dispensers drift over time in a downward manner. The Certus and the WD2 are using a similar dispense mechanism where you're pressurizing a bottle and opening up a solenoid valve to dispense. And the Combi is a peristaltic pump driven dispense mechanism. But largely you can see the same type of downward trend over time. This is normalized, and so the scale of this, you can see it's not a massive trend. Over 100 to a couple hundred plates, you might only see a few percent difference in volume. But that's something that we can be aware of, and that's something that we can control, actually. And so I'll talk a little bit about that. The other thing is having the integrated balances. It's really an insurance policy. So if a bottle is an ice bucket and it tips over, or if you run out of reagents, you didn't factor in the proper amount of dead volume, we have these systems notify us and stop running and be able to have an operator intervene to fix something and then to continue on. And so this is the balances are a nice feature, nice addition to the system. And so what we've done with Beckman and other people have done this with other scheduler softwares and integrated dispensers. as well, is to have the feedback loop and to have the software control dispense volumes. So in the case of the WD2s, we are able to use that information, feed it back to the dispenser, and as it drifts down, it bumps it up and keeps it dead flat for the course of a run. What I'm showing here is another reagent that we dispensed. This is actually filling up the octet plates that would be on the IgG system with about 32 microliters of buffer. So the octet's typically a 1 to 5 dilution of the supernate, and so we fill 32 microliters of buffer and then 8 microliters of soup. And so here we're filling just a bunch of plates to store in our refrigerator for future runs. And this was early days with this system late last year, and we were sitting there after the system was set up, and I was showing the WD2s to somebody, and they were talking about e-stops and how to control the system and how to respond to errors in the system. And right after it finished dispensing 32 microliters to the 11th plate, I said, well, let's see it. So it finished dispense, and I hit the e-stop right before it released the plate to the robot. And so on the SAMI side of things, you get a pop-up that says, do you want to ignore the error? Do you want to retry it? So just very quickly, the user I was working with hit retry. And so it already had 32 microliters per well on the 384 well plate. And so the retry is going to redo that dispense. And so now you have 64 microliters per well. And so then he was going around like, oh, we need to stop the screen, we need to fix things, and we'll start it back up, et cetera, et cetera. And I pulled it back and like, no, this is the beauty of what we just worked with Beckman to build into the system. Watch the system and just let it go. And so the next plate, obviously it thinks it overshot this dispense. And so the system undershoots the next plate, it overcompensates a bit. And then within a few plates, it dials itself back onto set point. And that's kind of shows the power of this. And so I think he was pretty impressed to see that, and then using this to show other folks of how we're controlling things is a nice example of it. Obviously, this is a gross example, and we typically would not have this much of a discrepancy in control. And oftentimes what people are going to talk about with a feedback mechanism like this, well, how can you catch a minor clog or a single tip when you're using a 8-tip or several channel dispenser? And so we determine the thresholds for what is an error. So usually you are setting your thresholds fairly tight around your set point. And so if you do have 1 in 8 tips clog, you're going to be 12% or so off in that dispense volume. So if you're dispensing 20 microliters, you're going to want to you want to probably keep that pretty tight or within a microliter, so plus or minus a microliter to, or maybe a couple microliters to be able to catch those types of errors. And so this is something we're pretty happy with and we like quite a bit. And so as we work with our scientists and we get protocols to run different assays, it would typically be something that looks like this. This is a generic protocol of dispensing cells, incubating, doing a transfer, further incubation, dispense, another incubate, and read. So what you typically hand over to your scientists is the reader data that comes off the end of this experiment. What we're able to share with our scientists now is every one of these incubation steps, we have a Gantt chart and we can show timing. And SAMI does that well, as well as several schedulers. But with the incorporated balances onto the system, not only can we show the Gantt chart that we hit all the timing points that they wanted, we can also say, well, you asked for 20 microliters, 10 microliters of antibody, the detection reagent. We can show exactly how much volume based on gravimetric results that these plates were receiving. And so if the data does have certain trends in them or patterns, then we're able to go look at timing as well as dispenser performance to be able to troubleshoot things. So it's been a nice addition to our systems. So this is a recent assay, and so we've done a lot of COVID-19 work over the last few months at Lilly, and a big part of that is running a significant number of ELISAs on this system. And so obviously this system would be very overkill if it were just performing ELISAs, and that's why we have so much equipment on here to do a bit more complex work, cell-based assays and high throughput flow cytometry. But this is something the system has been leveraged for, and it's been performing very well for us over the past several months. And so a typical ELISA process would be, do your wash and block, transfer samples, add your secondary, a substrate, and then in the system we do several OD reads. And that's one of the nice things is we can do several reads because the plates are always on there. You have a ballpark of, based on our scientists that we're working with, of how long it should take for development time. But since we have all the plates on there, it's an automated fashion, we can read from just a few minutes out to some of the plates we've read at 18 plus hours. And so it's nice to be able to just essentially queue up the system and let it go until these plates saturate, and then we back off and go to the time point or a couple time points before that when we're looking at to try to get good exposure without saturating. You do see a lot of steps in this process for a somewhat basic ELISA, and I would say that that is because of all the way steps. So every way step is a node, and then the script to analyze the way step is another node. And so before and after all these liquid handling steps, you're seeing a balance step followed by a script node. And so this method is significantly larger than you might see on another SAMI-based system. And then one of the nice things we're doing is our samples are coming off in 96-well format, and we're able to use the biomech. These ones are actually being normalized on the links prior to running the ELISAs. So we have a flat concentration of typically up to 96 samples in a 96-well plate. And then we're transferring into 3D4 ELISA plates, and on the I7 we're doing a 4-point dilution. So we're able to run dose response plates in a quadrant format in the by going from a 96 to a 3D4 well plate. And so we've generated results that look something like this. So the data coming off the system is we're using a DART report to aggregate the data coming off the system, and have made Spotfire templates that really, with minimal touch points, take that DART report and drop it right into a Spotfire template to be able to get what I would call kind of the 10,000-foot view of our systems. And so we're plotting row versus column and trellising by plate. So this is 2 sets of plates, A1 through 8, and then what we're calling B1 through 7. So what I'm typically doing here is sizing by the OD readout. And this is a 3D4 ELISA plate, and what you can see is, let me zoom in here on one of these, you can see a nice titration of some positive controls. So there's controls in the first column, and there are 2 fairly strong positive controls, and you see a nice titration. Again, these are sized by the OD readout. You see towards the end of the A plates and the last plate on the B plates, there aren't any samples in this portion. And so it's nice to see very consistent low sizing of the squares. And then again, I'm looking for streaks, edge effects, other patterns that might jump off the page to indicate a liquid handler issue in the system. And so this is really not the final analysis, but this is what I would throw data into a Spotfire report like this very quickly, or even as screens are running, just to be able to see any trends and make sure that the quality of the data is looking good. And then from this particular experiment, there are some sledgehammer, like really, really strong hits in plate A2 that ended up becoming important for us later on. And so from this system, I'm really trying to hit home the point that the functional highlights on here, we can run ELISAs, very wide variety and flavors of ELISAs, cell-based assays, high throughput flow, and then we can even prep plates for offline instruments. We have offline high content imagers, so we can do all the washing, fixing, staining of the plates on here and seal them up and prep them for a high content imaging-based assay. And so it's been a nice, a really nice system. Again, kind of jack of all trades on here, and we've got a lot of equipment, and we've really been focusing on ELISA the last few months, and so we're looking forward to returning to work soon and hopefully getting more cell-based assays. And late last year and early this year, we got a little bit of high throughput flow, some high throughput flow cell binding screens under our belts, and so we're excited to continue on that effort when we can get back to some normal projects. And so I touched on this whole process and going from single B cells to screening results to generate lead molecules. What I mentioned a little bit earlier when we were talking about the transfection system is, this is all in 96-well format, and the expression system, we're doing 1 mil samples. As we generate these leads and triage that list down, the samples do feed back in here a bit, and we decrease the number of samples, but increase the volume and the scale of those samples. And so we typically go into 24 oil plates up to 4 mil cultures. And at that point in time, we're typically not working with crude supernatants when we're going for our testing anymore. We'd like to purify that material. So early on, or last year, year and a half ago, we were scoping out a DNA purification system as well as a protein purification system. I think you can see here, we purify a lot of DNA. When we get to the protein purification side of things, we've down selected the number of samples pretty significantly. So it took a step back and tried to justify that we should spend the money to upsizing the DNA system and then do some small modifications on our existing systems to enable the protein purification side of things. And so that's where I'm going to talk about those little tweaks that we've made recently and how we can enable the throughput that we need for protein purification. And I've already shown why and how we've upsized the DNA purification system. So kind of reallocating the money where we get the best bang for our buck. And so this is the Beckman Quant and cherry pick system, again, what we call the IgG system. And so as we pellet cells and harvest the supernatants and we measure the titer on the octet, well, we can run 24 well plates on the system in largely the same manner. But what we can also do is use the fleet of liquid handlers on here to add resin to those supernatants. And then we seal the plates up and then put them on the bioshakes for roughly 30 minutes or so. Then what we would do is pellet those beads of resin in the high-G centrifuge. And then where you typically are having roughly 4 mils of sample and resin in the plates at that point in time, take off you've pelleted the resin, so it's in a pellet at the bottom of the well, take off the roughly 3 mils of supernatant remaining on top of that. So what you have as an output of this system is around 1 mil of liquid and resin in these 24 well plates, and the antibody is largely bound to the resin at that point in time. And we've done Mike Cohen's done a nice experiment showing that we are capturing nearly all of the antibody in the supernatant. And so from here, we can go to the DNA purification system where we have the new i7 with a couple positive pressure devices and use the i7 to transfer those roughly 1 mil of sample and the resin into 96 well filter plates, and then use the positive pressure devices to push the sample through the filter plates, do washing, elution, and then use the Biomech to make an aliquot. Again, read it on the integrated spectrophotometer, and then have the i7 take a work list in and break out those samples by scientist or requester or user. So from here, we will measure the amount of material and then break the plates out and give people plate maps of their purified material. And so again, putting a few bioshakes on another system and a couple positive pressure devices on this guy and using all the other equipment that we already have in place really is a little bit of equipment to enable a whole nother automated process that we weren't doing previously. And so again, this is the overview of the process. And as I talked about in the very beginning, a lot of this is tied together with a LIMS infrastructure, and a lot of folks have been helping on that front. So it's a huge effort, not only to build these systems with our team and the collaborators such as Beckman and Fujifilm and Biocero. It's been a lot of work with the informatics folks and the IT folks to develop the infrastructure, the LIMS infrastructure for these. And so from this whole process, and the whole reason why this all exists is to really deliver multi-species ELISA data, cell binding flow data, functional cell assay data. And then when we purify DNA back on the DNA purification system, we do make a cut for sequencing as well. And so by the time you have your screening data coming off the right side of this process, you have sequencing results as well to tie that back to. So it's really a rich data set to give our scientists as much information as possible to whittle down our lead molecules. And so with that, again, it's a really huge effort. I joined a couple of years ago, so I'm very thankful to have the pleasure of sharing the work from the group. But there's, Tom, Melissa, and Mike had a huge role in this for several years before I got here. And David, Liliana, and Andrew are in my group and help with the automation side of things as well. And so everyone in my group is Mike, David, Liliana, Andrew are responsible to support and keep these systems running and work with Beckman and the other vendors to improve these systems and evolve them over time. And our scientists that carry out the single cell process, our screening group, as well as our sequencing folks, it's a huge amount of people in what we call the automation group, all underneath Tom, that use these systems, have suggestions, help evolve these systems and improve them. And then the scientists we work with upstream and downstream of us, as well as the IT folks, have a huge role in this. And then we have several lab managers that help support what we do, that we're very thankful for, and the management support to be able to build out this infrastructure. And again, the people we're working with at Beckman, Biocero, and Fujifilm to help develop these systems and to continue to improve them over time, incorporating balances, integrating new devices as they come out, exploring new technologies and workflows. We have a lot of stuff to work on as we continue our process and look for new opportunities to incorporate new technologies, whether it be biology-driven or automation-driven. So it's always it's a never-ending cycle of improving these guys. And so with that, I'd like to thank everyone for their time and open it up for questions.
Thank you, Paul, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. Our first question is, why not TR-FRET instead of Elisa?
Thanks. So the question about FRET versus ELISAs are, we have a lot of scientists here that have been doing things a certain way for a long time, and so the ELISAs are kind of what we lean on. And I would say we have a vast amount of expertise in the ELISA area, as well as the costs associated with them. So they're fairly inexpensive to run, and we can develop the assays very quickly. And we, like I mentioned in the presentation a little bit, we run a variety of formats from antigen down to capture formats to multi-species to 4-point, 8-points, replicates, and whatnot. So we can develop these quickly and deploy and run them in a very fast manner with minimal assay development time. And so that's typically our first pass is going towards ELISAs.
Thanks, Paul. Okay, our next question, this is a two-part question, says, can you view information and data in real time? And what are you using to push and pull data to your different systems?
Yeah, that's a really good question. So it is system and vendor specific. So on several of the systems, we have scripts where we are interacting with our LIMS system, where we can work with the vendor in some instances on a lot of the Beckman systems, where we can ping our LIMS system to gather what information is what samples and concentrations and other data for those plates that are coming onto a system. The system can process plates, do transfers, run screens, and then when it's typically done, it will it also has scripts to push information back to LIMS system to say samples move from this barcode and well on this plate to this new plate, and now this is data associated with those samples. And so several of our systems have things set up that way, whether they're from the Biocero side of things, Beckman side of things, or the Fujifilm side of things. So we kind of are working with the vendors on each platform to accomplish the task of integrating with our LIMS systems. And then the part about, the second part of that was looking at data in real time. And so the main ones we're looking at real time are actually all the Beckman systems, and that's where we're using DART. So that's basically a database and a nice GUI that sits on top of the systems to be able to look at what's running on the platforms. And that's more in a table format. And so what I talked about on the assay system is I'm taking that DART report and then trying to get that into a visualization platform like Spotfire. Is kind of what I tend to lean on. And so we're trying to marry those two things. So being able to very simply create DART reports and have a lot of predefined DART reports, and I can export and link right into Spotfire templates that I've created to get a quick view either during runs and after runs especially as well.
Thanks, Paul. Okay, your next question, what different B cell screening approaches do you use?
That's probably a more upstream question than I might be able to answer. So we have several types of species, and we have developed different immunization, like animal-based immunization strategies. We have, as I mentioned, a number of other types of antibody discovery platforms, not just B-cell discovery platforms. So I don't know that I can go too much into the different platforms upstream of us very much in this talk.
Thank you. Now this user wants to know, is SCP safe?
I'm not exactly sure what they mean by SCP.
Right. So let me see. Let's see if this user, if you want to go ahead and just retype your question and maybe elaborate a little bit more, we'll try and get to it before the end of the presentation. But for now, let's go ahead and move on to this next question. How do you analyze complex data sets such as flow cytometry data?
Yeah, that's a really good question. So the DART reports are really helpful for taking reader data that's kind of a flat file coming off the system. When you have something significantly more complex like flow cytometry data, that's where we need to develop a different type of work stream to be able to use third-party software such as FlowJo to be able to take the data coming off the system, process it in FlowJo, and then pop it into something like Spotfire to visualize your results. And so that's where I've basically written a script to streamline that process. So if we have a FlowJo template, FlowJo can be run from a command line. And so essentially you can write a script to point FlowJo to all the data and to feed FlowJo a plate at a time and consolidate all those results into one large CSV file. And then that's the process data that you can then put into Spotfire or a downstream analysis toolset. So it's basically another instance where we kind of had to create a little intermediate tool to take the data coming off the system and get it into an analysis platform like FlowJo to be able to properly process the data and then be able to put it into a visualization and downstream tool that our scientists are comfortable with using.
Okay, we have time for a few more questions. Let's go with this one. What additional system or systems do you feel would be compliant with this workflow?
Yeah, to complement the workflow, I think they were meaning.
Sorry, yes, complement.
No, it's okay. Yeah, that's all right. So what we have on this whole platform, I don't know that we're going to build out several or any more new big systems. I think we want to look at ways to leverage the existing system. So I talked about enabling protein purifications with some fairly small upgrades to the existing platforms. So I don't know that there's going to be a lot of hardware changes. I think software is really where we're going to be able to gather a lot more information on there. We have balances on a lot of the systems. We put in a camera system late last year to basically have eyes on all these systems. And I think one thing that was interesting with that, the first night we installed the camera system, we caught a bug on a system that is very infrequently occurring. And we were able to take the data logs from the system as well as the video and hand to a vendor to show them that, this is what the software is reporting, and this is what is actually happening with cameras. And so deploying cameras, balances, capturing more data, we want to look at every system and try to find where the weak points are and make them more reliable, as well as try to look to continue to find software solutions to be able to streamline our process and make things more efficient. So, may not build a new big system. I would say one thing that kind of would help feed a lot of these systems would be some automated tissue culture. We have a selectee that's quite old, and that might be one system that would really help complement what we're doing here. So we're thinking about that, but we have no plans right now to be able to pull the trigger on any of that. And so I think that's the main thing that we're looking at is possibly something in that space, but a lot of what we're doing is going to be more on the software side of things.
All right, we have time for just one more question. Where do you see these automated platforms going over the next 1 to 2 years?
Yeah, that's a good question. So we've been working on the LIMS side of things significantly. So connecting these more, getting things streamlined. So even our standalone Biomix, I talked about the transformation step largely being a manual process. So hooking up a lot of our standalone equipment, getting barcode readers on there, and connecting the standalone equipment and the integrated systems all to our LIMS platform. And then, I don't know where things will happen if there's ever a possibility of connecting the systems and somehow, mobile robots and things like that have been talked about the SLAS conference. This might be an opportunity to leverage things like that. That would be much further down the road, but we have a lot of islands here, and a lot of the touch points in between the islands are typically storing plates in 4-degree refrigerators. So I would say at some point in time, you can envision a process like this. There might be a way to connect them with a mobile robotic platform. But that's pretty far down the line, I think, not in the next year or two.
Thank you, Paul.
Okay.
Do you have any final comments for our audience before we close out today?
No, I just want to thank the vendors we've been working with, and a lot of the people in the automation group. Like I mentioned, I've been here a couple of years, and so there's a lot of people that have been working on this for several years before I joined, and it's been a pleasure to work with everyone on the Lilly side and on the Beckman and Biocero and Fujifilm side as well.
Great. Well, thank you so much for your time today and for your research. We'd also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for today and for their interesting questions. Any questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care. Bye-bye.
Thank you.